Hi everyone, today I'm going to present a paper titled PS3, a multimodal transformer integrating pathology reports with histology images and biological pathways for cancer survival prediction. Starting off with the introduction and motivation behind this method. While multiple methods have integrated whole slide images with genomic or transcriptomic data for survival prediction, few methods have explored the idea of incorporating pathology reports into survival prediction workflows. This is in spite of the fact that pathology reports represent a readily available source of medical knowledge and are routinely prepared as part of standard clinical and diagnostic workflows. Moreover, they contain expert-derived diagnostic insights and can complement histology and genomic data. In this method, we propose to integrate complementary information from all three modalities: whole slide images, genomic data, and pathology reports for survival prediction. One of the major challenges associated with this multimodal fusion is differences in the scale and structure of data across the three modalities. To address this, we propose to create representative embeddings or prototypes for each modality to create more balanced representations and enable more effective multimodal fusion. Now I will discuss how we create prototypes for each modality. For genomic data, we multiply our gene expression vector with binary pathway composition vectors. This gives us a vector for each pathway consisting of the genes involved in that particular pathway. We pass these vectors through MLPs to create standardized pathway prototypes. Secondly, instead of representing each whole slide image as thousands of individual batches, we leverage the morphological redundancy inherent in whole slide images to cluster their batches into a set of histological prototypes, each representing a recurring tissue pattern. We use Gaussian mixture models to create these histological prototypes where the mean represents the prototype features, the covariance represents how much the prototype varies, and the weight represents how common the prototype is in that whole slide image. Given a single pathology report, we split the text of the report into text segments to create feature embeddings of the report. Given a batch of variable length pathology reports and consequently variable length report embeddings, we pad them to the length of the longest report in the batch. We aim to standardize the report representations while preserving the most relevant information. To achieve this, we employ transformer-based self-attention. We project the padded report embeddings into key, query, and value vectors using linear learnable transformations. Then we compute a single importance score for each segment of each report by averaging the attention weights across all query positions to show on average how much the entire report attends to each segment. We then use these to rank the segments and select the top NT segments to create our diagnostic prototypes from our pathology reports. We project the prototype representations of all three modalities into keys, queries, and values using learnable linear transformations, and use them as input to our multimodal fusion transformer. The transformer consists of a total of 9 attention mechanisms, including 3 self-attention intra-model interactions and 6 cross-attention or cross-model interactions between the three modalities. These attention scores are then multiplied with the value representations and then averaged within each modality and concatenated to form the final fusion multimodal feature vector, which is used for survival prediction. We have compared our proposed approach, PS3, which stands for Predicting Survival from 3 modalities, with a clinical baseline as well as multiple unimodal and multimodal baselines, across 6 cancer types from TCGA. We note that PS3 achieves the highest overall performance and is among the best performing methods in 5 out of the 6 cancer types. In addition, we have also conducted multiple abelation studies to assess how performance varies across different model settings. We visualize the histological prototypes created from the whole slide image and the cross-attention interactions between the different modalities. Specifically, we illustrate a whole slide image, a heat map showing the spatial distribution of patches corresponding to each prototype in the whole slide image, the proportion of patches assigned to each prototype, and representative patches from the most significant prototypes. We have also visualized cross-attention between the modalities, specifically pathways and pathology reports to histology and histology and pathology reports to pathways. We have also generated word clouds consisting of 2 word phrases from the pathology reports for high-risk and low-risk patients. Additionally, we also generated KM curves comparing the proposed method PS3 against other multimodal baselines. The high-risk or red and low-risk or blue groups were stratified using the median predicted risk value. I hope you enjoyed this presentation and found it helpful. Thank you for listening.